Hello. Okay, today we'll be talking about animal disease models. We'll be talking about what they are, why they're useful to us, um, what are they used for, some methods of generating them, which is pretty cool, pretty awesome, and how we can develop therapies from this, which I'll go over in a future video. So, an animal disease model is just a system that mimics a type of human disease in another organism. So this gives you insight on the disease mechanisms, the potential therapies, and disease origin. Let's say you do just a normal EMS mutagenesis to get your candidate gene, like in Drosophila or something, let's say. There's varying types of models that exist, but today we'll be looking at in vivo, which is whole organisms ones, which is using whole organisms to study diseases. There's like in vitro, ex vivo, which is just taking tissues out of an organism, and then in vitro is specific cell lines or culture lines. So some guidelines need to be in place if you're injecting a bunch of animals with diseases. I think so. And this one that's used often is the 3R model. So replace animals that you don't need to use. So what do I mean? If you want to model a disease in chimpanzees, but you can just as easily model that disease in a fruit fly, you know, go ahead and use the fruit fly instead of the chimpanzee. Reduce the amount of organisms that you need to do. Let's say you're doing an age of onset study where you're seeing how the disease progresses through generation to generation. Use the least amount of generations. You don't need to use that many. And obviously refine your process before you do this. You don't want your animal to suffer for no results whatsoever, and you want to suffer as little as possible. Um, at least I hope you do. One organism that we use is the mouse. I love mouse. There's Remy from Ratatouille. Their advantages are 99 of their protein coding genes are shared with us, shared with humans. That's the glory of evolution. Uh, Genes are highly preserved. That's why we're 50% like a banana. So this means any disease that we use in this model is pretty translatable to humans. Um, their behavioral traits can be observed. They are quite socially intelligent, and they're well characterized in the scientific world. Like we've been using these guys for ages. I love mice. I love rats too. You should love rats too. They have a double standard for them. Then disadvantages are obviously ethical concerns, and they're pretty expensive to maintain, and you have a bunch of rats in your laboratory. The next organism we use is zebrafish. These are pretty interesting as they have lots of progeny and they have a transparent embryo. What this means is you can more closely study congenital disorders that are present at birth or very near birth. And also, 70% of coding genes shared still, so still pretty translatable to humans. The disadvantages are they have a pretty long lifespan, expensive upkeep, and you have to have an aquarium in your laboratory if you want to study them. And then we have fruit flies, Drosophila, the E. coli of animal disease models. They have the highest number of progeny, they're the least expensive to upkeep, they have little to no ethical concerns, and they have very short life cycles. All of these are very great points. Why don't we use these guys forever? It's because they have no real observable behavioral traits. They just sit and fly. They don't really do anything else. And they're the least comparable to humans genetically. So if you find a therapy for a disease in Drosophila, then immediately apply that to humans. That would make you a bad scientist. So there are 3 different ways to generate a disease model. Not really, there's more, but these are what we'll talk about today. So we'll talk about transgenic overexpression, homologous recombination, and CRISPR-Cas9. Whoa, big name. Transgenic overexpression is normally for gain-of-function diseases, for modeling gain-of-function diseases. For example, if they have a dominant negative effect, where the mutated allele's protein product interferes with the wild-type allele's protein product. Homologous recombination, we use that for loss of function in knockout events or single nucleotide polymorphism diseases, which are usually recessive. Both of these are recessive. Why? It's because if we only have one copy of the allele, is normally haplosufficient, right? So enough of that allele's product is required, can have a enough of that allele's product is good for normal function. But in this case, it's haploinsufficient, and so only having one copy of the allele is not enough for normal function. And then we'll see CRISPR-Cas9, which is bombshell. Very cool, very cool. So we'll look at transgenic overexpression, sometimes called ectopic overexpression, if it's expressed in a different tissue than it originally was, the disease or the gene of interest, I mean. So a transgenic construct is created where you have your transgene, your gene of interest, and then upstream of it is a promoter sequence. It doesn't have to be specific, tissue specific, but there is a promoter sequence there, which helps you regulate the expression of this gene. And normally a marker is there as well, to select for if this transgenic construct actually integrated into the organism. How do we do this? It's normally pronuclear microinjection. So this construct is injected into a male pronucleus and then is combined with female oocyte, which makes us a zygote. And then the integration occurs randomly whenever nicks in the DNA occurs. So whenever any DNA damage occurs, boom, transgenic construct, boom, gets in there. The problem with this is we don't know how many copies of our transgene are in there, so it can affect like gene dosage. We don't know when it will happen. Like this is not very specific science. So it can occur later in development, which can cause mosaicism, where the mature organism has cells without the transgenic construct and cells with the construct, and so it just, which is not really a bad thing. It may make it easier to study even by comparing it with the non-affected cells. And it can also cause insertional mutagenesis, where the construct inserts itself in between a coding sequence, and that messes up the protein, which may be vital for normal function, let's say. Yeah, so there can be different promoter sequences. You can have ubiquitous ones, which just express your gene constantly, no matter what. Inducible ones with heat shock, so it is only expressed when high heats. Tissue-specific promoter sequences, we have this all over in our body, right? We have, or else we'd all just be one blob. We wouldn't have like different limbs or different organs. Everything would be expressed all the time. And then we have TET-off, TET-on system, and binary expression, which I'll explain in the next slides. So TET-off, TET-on, what is it? It includes the PTET promoter. So you put PTET promoter upstream of your gene of interest, and then tetracycline transcriptional activator would also be present, which binds to PTET at baseline, which drives expression at baseline. So in its natural state, TET-off drives transcription, therefore drives expression of your gene. But as soon as we add DOX, which is a tetracycline analog, the reason we use DOX is just more stable. This inactivates this activator. It's a funny way, inactivating the activator. And so this immediately turns off transcription of our target gene. And it is very fast to do this. Like as soon as that DOX gets into the organism, Normally we just feed it to the organism of choice. As soon as it's introduced, it binds, inactivates transcriptions, cool. But for it to actually, for transcription to come back on, and this docks to like flush out the system, it takes quite a while. So that is its downside. And then the opposite is TET-on, where it's reverse TTA, right? At baseline, expression is repressed. At baseline, there is no expression until doxycycline, which again, tetracycline analog is introduced, where transcription of your gene of interest occurs. And again, this is fast to switch on, this is fast to switch off. Choose whichever one you want, depending on your experiment. Then we have GAL4/UAS, so binary control, which is sort of similar to this, I'll show. So GAL4 is a transcription factor, and then you put this under a tissue-specific regulatory region, and you create a line of GAL4 mutants that only express GAL4 in a specific tissue. Let's say in your heart, it only expresses GAL4 in your heart. And then you create another line of mutants where your gene of interest is under the control of an upstream activating sequence. And then you have your two mutants and you crossbreed them. And then this mutant has your gene of interest in all of its cells, but is only activated by GAL4 in a specific tissue. And this can give you ectopic expression of a mutant gene in a different region, which is very useful. The reason why you do this is because some diseases may be fatal, cause progeny death, so you can just keep crossbreeding. I know it sounds like a bit, again, ethical concerns, but you can just keep crossbreeding, and then the progeny can be studied indefinitely. Next, we have the second way of creating an animal disease model, which is homologous recombination. What is it? This uses recombination events, which are really rare. Normally, it's just sister chromatid exchange, right, during meiosis, right? The same parts of the DNA exchange information. And this causes deletion/disruption of the genes, which you can model loss of function for. Or you can introduce mutated genes in your specific locus. You do this by extracting embryonic stem cells in the early blastocyst stage. You manipulate them ex vivo, and then you replant them into a surrogate host, so that all of your cells contain the loss of function or the mutated gene. So first, you have to make a targeting vector, and this is made by, first of all, finding a good selectable marker, in this case, NEO-R, with regions of homology that are the same as your target locus. So here's your target gene, here's your exon 1, exon 3, copy that to your targeting vector, and have it flank this selectable marker here. And then in addition to that, since recombination events happen naturally anyway, you have an outside marker, HSV-TK, that is present for negative selection. So normally, if this homologous recombination is successful, thank God, here exon 1 exchanges, and then exon 2 is replaced with NEO-R, and you can positively select for it. If that doesn't happen, let's say this targeting vector recombines with a totally different part of the chromosome, it will have this HSV-TK gene on it, right? And if that's combined with ganciclovir, it causes cell death. So yeah. And then if you want to introduce a mutated gene, just again, create another targeting vector with a mutated gene, replace NEO-R with this targeting gene. So next we have Cre/LOXP gene inactivation. which is kind of just another version of homologous recombination. The way this works is because it uses Cre, which is a site-specific recombinase that recognizes LOX-P sites and combines them if they are in the same orientation, whilst excising this sequence. Look, it loops and then cuts and then combines both LOX-P sites. Very cool, very cool. So again, you create another line of mouse with LOX-P sites flanking the locus, and then you create another line of mouse that express Cre only in specific tissues using a specific tissue promoter. Crossbreed these, and then the mouse progeny only exhibit Cre recombinase events in a specific tissue because Cre is only expressed in a specific tissue. Again, you can combine this with inducible expression systems like TET-on, TET-off to control both the timing and the location of your gene of interest being expressed. Very cool, very cool. And then the final way of creating an animal disease model is CRISPR-Cas9. How this is done is Cas9 induces a double-stranded break after it's been recruited to its target sequence by its intrinsic guide RNA, this sgRNA here, this guide RNA. And then the most common mechanism of repairing double-stranded breaks in organism is non-homologous end joining. And this causes indels to occur because it is not accurate at all. The cell has no idea what sequence was in here before the double-strand break. So the insertion deletion indel may occur. Models loss of function, can model loss of function disease. And then if it's homologous direct repair, it can actually revert your mutated allele back into its normal, because if it uses an analogous chromosome with the normal allele, it can repair it, right? So normally your mutated allele was here, wild type is used as a template. So now you have both wild type. So that is a form of genetic therapy which can be used. This is easy and efficient to do, but it can cause off-target effects. Like this guide RNA, anything that matches even closely related to it, or double-stranded break, double-stranded break, double-stranded break, all over the genome, all over the GAF. You can also have two guide RNAs that can be injected to mimic deletions. So two guide RNAs, one would cause a double-strand break here, another would cause a double-strand break here. That middle part is excised. So how do we generate therapies from this? Again, you can get candidate genes from simple EMS mutagenesis experiments. You can do a wide range of chemical strains. They use this often. Drosophila have the most progeny lowest life cycle, right? So just throw some chemicals at them, see what works, then test it on mice, then test it on, then do some clinical trials on humans. The life cycle of disease can be more closely investigated since we're using these organisms that have a much lower life cycle than humans. The effects of the disease on someone's life cycle can be observed, and then therapeutic strategy effects can also be clearly observed. So let's say one of your therapies is using an integrating viral vector, insertional mutagenesis, which activates oncogenes, which causes cancer, can be observed within other organisms before doing it on a human, preferably. Don't give people cancer, preferably. Anything else I want to say? No, I think that's it. Thank you for watching. I hope this was clear somewhat. Bye-bye.